Hey guys, so this is part 2 of our photosynthesis series. This one's going to go over the Calvin cycle, also known as the light-independent reactions. The first couple of slides will be a summarization of some of the main concepts that we went over for the light-dependent reactions, and then we'll segue into the Calvin cycle. So here we go. All right, so I don't know if you're familiar with the comic strip Calvin. It's like a comic strip about this tiger and a little boy. But it's not this Calvin, it's actually this Calvin. The cycle was named after this guy. I can't remember his first name. Obviously, it was never that important to me to remember it to begin with. But he's the one that kind of discovered and constituted the Calvin cycle as we know it. So here we go. So don't forget, if you're a plant, you still have a lot of needs that animals will have. You have to be able to get organic molecules, but you're going to be producing those yourself. And it's going to be the same organic molecules that we talked about when we talked about biomolecules. Those are our carbohydrates, our lipids, our proteins, and our nucleic acids. Somehow you're going to need to be able to store chemical energy, at least short term, until you can use it for the Calvin cycle. Don't forget, when it comes to making these organic molecules, ATP is made in the light reactions, and then we have to store it so that we can transport it to be used in the light-independent reaction so we can actually make our organic molecules. So we have a lot of similar needs, just like you would if you were an animal. You still need organic molecules, you still need to store chemical energy. Okay, so our light reactions are all about obtaining light energy in the form of solar energy from the sun, and using that light energy to make two things, to make ATP, which is going to be our storage molecule, and NADPH, which is our electron carrier. So now that we have these two things, now that we have ATP and NADPH, we can actually build our organic molecules. So we're getting ready to do the synthesis part of photosynthesis. So we want to make glucose, C6H12O6. So let's think of what we have available. We know we have energy. We know we have reducing power in the form of our electron carriers. The raw materials we're going to use for this part of photosynthesis is going to come from carbon dioxide, or is going to be carbon dioxide. So we're going to take that carbon dioxide, we're going to take the carbon out, and we're going to fix that carbon into compounds that we can use to make glucose. So let's think about it. Now, remember, photosynthesis is almost like the opposite of cellular respiration. In cellular respiration, we started off with our glucose molecule, and we oxidized it all the way down into carbon dioxide. In photosynthesis, at least in the Calvin cycle, we will start off with that carbon dioxide even though it is fully oxidized, and we will add energy to it to make it glucose, C6H12O6. So carbon dioxide is really, really oxidized, whereas glucose is really reduced. We're going to synthesize, which means we're going to build something. In order to build things, you're going to have to hold those things together with bonds, which means you're going to have to put energy in to create a bond. So it makes sense that we're going to start off with something that doesn't have a ton of energy. Okay, so that there's not that interruption, there's not that interference. Now, cellular respiration and even the light reaction or the light dependent reactions of photosynthesis were both downhill processes. We started off really high energy, and then as those electrons moved, they lost some energy in the process. Now, the synthesizing section, the Calvin cycle of photosynthesis, is going to be the opposite. It's going to be an uphill series. We're going to start off very low energy, and we're going to keep adding energy to that every step, until we end up being high energy. Just like everything else we've talked about, this is all done by in a series of steps, and it's all catalyzed by specific enzymes. Now, you're probably wondering, okay, if we're going to build these bonds, we need energy to do it. Where does the energy come from? Well, we already made it. Remember, in the light dependent reactions, we made ATP and we made NADPH. Actual energy and reducing power. So we're going to use the energy stored in these two molecules to create these bonds to make glucose. So let's think about where all of this is going to happen. We're still in the chloroplast, but the Calvin cycle happens in the stroma of the chloroplast, so the liquid section of the chloroplast. Remember, light-dependent reactions happen in the thylakoids or thylakoids, and the Calvin cycle happens in the stroma, in the liquid regions. What do we need to start? We need ATP, which we've made. We need NADPH, which we've made. And we also need an input of carbon dioxide, which we can get as long as the stomata of our plant leaves are open. Excuse me. So this is an entire showcase of the Calvin cycle. And this picture makes it look really, really busy, but we're going to step through it together and make it work. Now, in cellular respiration, we had the Krebs cycle, which looks very similar to this. The Krebs cycle was 2 complete turns, 1 for every molecule of acetyl coenzyme A that we produced. The Calvin cycle actually turns 3 times, 1 for every molecule of carbon dioxide that our plant can take in. So the Calvin cycle is a 3-turn cycle. This is going to happen 3 individual times. And it's divided into 3 steps. The first step is called carbon fixation. The second is called reduction. And the third is called regeneration. Just like how the Krebs cycle had intermediates that we started off with and we added things to, the Calvin cycle works the same way. Our first intermediate is a 5-carbon compound called ribulose bisphosphate, or RUBP. When we do carbon fixation, we're going to take the carbon away from the oxygen and add it to ribulose bisphosphate. So we're going to end up, for every turn, with 1 molecule of what is now a 6-carbon compound. All right, so that's why it's showing you in multiples of 3. This is for turn 1, this would be turn 2, that would be turn 3. The enzyme that does this fixing of carbon is called rubisco. Rubisco stands for ribulose bisphosphate carboxylase. So it tells you that it utilizes carbon. It cuts carbon away and adds it to something else. Now, once we have our 6-carbon compound, we're going to go ahead and cut those in half all the way down the middle pretty much, like this. And we're going to end up with 6 molecules of a 3-carbon compound called PGA, or phosphoglycerate. Now, once we have our PGA, we're going to do a little bit of rearrangement, and we're going to make a different kind of 3-carbon compound. To do that, we're going to utilize some of this ATP that we've already made right here. So we utilize 6 molecules of ATP. That's why we're starting with ATP and ending with ADP. Then we have to go ahead and reduce those carbons. If you think about it, glucose has a lot of hydrogens. It's C6H12O6. So we have to put some hydrogens into the mix somehow. Well, we stored a bunch of hydrogens in our electron carrier known as NADPH. So in our second step of the Calvin cycle, we're going to take those hydrogens away from NADPH and add them on to these carbon skeletons that we've made. Okay, so we utilize 6 NADPHs in the reduction phase. Now, once we have our carbons that have some hydrogens on them, we've now made a molecule, it's still a 3-carbon molecule, that's called G3P, glyceraldehyde 3-phosphate. You've seen this before. This is the same G3P that we utilize in glycolysis, or that we make in glycolysis. So this is the ending step, really, the main reason, I should say, for the Calvin cycle. It's to make this G3P molecule, because G3P is the precursor used to make glucose. And from glucose, we can make sucrose and cellulose and starch and whatever else the plant needs. So the whole point of the Calvin cycle is to make G3P. Once G3P has been made, the remaining carbons are going to be regenerated into ribulose bisphosphate, and to do that we're going to utilize 3 molecules of ATP. And that, in a nutshell, is the Calvin cycle. So let's go over. It turns 3 times. We're utilizing carbon dioxide. We're taking the carbon away from the carbon dioxide and adding it to an intermediate that already exists called ribulose bisphosphate. That that step is catalyzed by the enzyme Rubisco. We're going to go through and change up the structure of our what is now a 6-carbon molecule a couple of times. In that process, we're going to use some ATP. And then we're going to add some hydrogens to those carbons. We're going to take those hydrogens from NADPH, and that last step is going to make us our precursor molecule for glucose, which is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is then going to be removed from the cycle and sent wherever else to make glucose, and we're going to regenerate ribulose bisphosphate, that intermediate that we start off with, using 3 molecules of ATP, and the cycle will repeat itself. Okay, so like I said, same G3P that we used in glycolysis. So, face. So let's talk about glyceraldehyde 3-phosphate. It is the end product of the Calvin cycle, and it is 3 carbons big. It's very energy rich, and it's the ending result. So when we end up, or when we produce glyceraldehyde 3-phosphate, this is known as C3 photosynthesis. So the C3 comes from 3 carbon. This is what most plants do. This is the photosynthesis that you've been taught since 7th grade, but it's not the only kind of photosynthesis available. We'll talk about what some alternatives are in class on Monday. Or if you're 1B, I've already seen you, and you talked about that on Friday. So glyceraldehyde 3-phosphate is a very important intermediate. Without G3P, we couldn't make any of these things. We couldn't make glucose for carbohydrates, or lipids for our waxes, our fats, and our phospholipids, amino acids for our proteins, or even nucleic acids for our DNA and RNA. So let's talk about Rubisco a little bit. Rubisco is a highly important enzyme. It's the enzyme that takes the carbon out of carbon dioxide and fixes it into an intermediate that we can make G3P from. Again, Rubisco stands for ribulose bisphosphate carboxylase, and it's one of the most, if not the most, abundant enzymes in the world. Every single plant has tons of these enzymes. So let's go through an account. 3 turns of the Calvin cycle give you 1 molecule of G3P. So 3 carbon dioxides give you 1 G3P. You remember, carbon, carbon, and carbon. All 3 of these carbons are coming from this carbon dioxide. So 6 turns of your Calvin cycle is what it will take to build 1 molecule of glucose, because you need 6 carbons. You need 6 carbon dioxide molecules to give you enough carbons to make glucose. So 6 CO2s equals 1 glucose. It also takes 18 ATP and 12 NADPHs for this to happen. So in terms of energy, 18 ATP and 12 NADPHs give you 1 glucose molecule. If you ever made extra ATP, like for example when we go through cyclic photophosphorylation and we are continuously making ATP, any of that leftover ATP can be used somewhere else in the cell. So let's summarize. We know we have our light-dependent reactions. They produce ATP, they produce NADPH, they utilize water or consume water to do this, and free oxygen is produced as a byproduct. Then we have the Calvin cycle, also known as the light-independent reactions. It consumes carbon dioxide, it produces G3P, which is our precursor for glucose, it regenerates ADP. Another way to think of this is it uses ATP. So let me write that in for you, because that's a simpler way of thinking about it. So it uses ATP, and it also uses NADPH. Okay, so again, let's go over the Calvin cycle. It consumes carbon dioxide, produces G3P, which is our precursor for glucose, uses ATP, and uses NADPH. So this whole process, all of this, that's photosynthesis. So again, light reactions. They happen in the thylakoid membrane. They depend on the energy from sunlight. Electrons are going to be, water is going to be taken up by the root. That water is going to be split into oxygen and hydrogen. The oxygen will leave and exit the leaf via the stomata. The hydrogens will have their electrons removed from them, and the hydrogens will be pumped into the thylakoid space, creating that gradient that we are so familiar with when it comes to the electron transport chain. There are two sets of photosystems located in these thylakoid membranes. Photosystem 2 is first, which is P680, and photosystem 1 is second, which is P700. In those photosystems, we have a bunch of accessory pigments, and it's their job to gather sunlight and pass that sunlight on into the reaction center of the photosystem, and that's where chlorophyll A and B reside. Those electrons that we took from the hydrogens are also in that photocenter. When that energy gets to the photocenter, the electrons get excited, they change state, and they flow from protein to protein downhill, and we make ATP from that. That is the first part of non-cyclic photophosphorylation. So we've just made ATP. We went from photosystem 680 to we've just arrived at photosystem 700. Same thing is going to happen all over again. Sunlight's going to come in, accessory pigments are going to collect that energy. Energy is going to be passed to the reaction center. The electrons are going to be excited. They're going to change excitation states. They're going to have more energy. They're going to flow downhill. And at the last part of that process, they're going to couple hydrogens to NADP, making NADPH. That's the light reactions. In the dark reactions, the ATP and the NADPH that we made in the light-dependent reactions are going to enter the stroma of the chloroplast, which is where the Calvin cycle takes place. The stomata have been allowing carbon dioxide to infiltrate the leaf. Those carbon dioxide molecules are going to be what we use to run the Calvin cycle. The enzyme Rubisco is going to strip carbon off of the carbon dioxide and add it to a 5-carbon intermediate called ribulose bisphosphate, or RUBP. Through a series of steps, we are going to take that precursor, we're going to break it up into 3-carbon compounds. That's going to take some energy, so we utilize ATP for that step, 6 of them to be precise. Then we will take those same molecules and add some hydrogens on them using NADPH to do that. So we've utilized ATP and NADPH. And then we're going to make the compound G3P, which is a sugar. It's the precursor that we're going to use to make glucose. This whole process happens 3 times so that we can make 1 molecule of G3P. To make 1 molecule of glucose, the process has to happen 6 times in total. When we utilize ATP and we utilize NADPH, we've now oxidized them. So now we have ADP and NADP. We're going to send those back to the light-dependent reactions so that we can remake them again. So we can make more ATP and more NADPH. Right? So this is how plants make both energy, that happens in the light-dependent reactions, and sugars, which happen in the light-independent reactions, or the Calvin cycle. So this is the simplified version of photosynthesis, just like this is the simplified version of cellular respiration. The two go hand in hand. It's a big, can't think of a good description. It's a big passing game almost, because the products of one become the reactants of the next, as you can see. We make glucose and oxygen in photosynthesis, and those are our products. But in cellular respiration, we use them to make ATP, carbon dioxide, and water, which become the products of photosynthesis. So one can't exist without the other. Big circle of life thing. All right. If you can answer these questions, this is our summary of photosynthesis, then you're in good shape. You have a good understanding of what's going on, at least a bare bones understanding. We'll talk more about these in class. All right, just to give you an idea of how this affects our world, on a global scale, photosynthesis is one of the most important processes, if not the most important process for the continuation of life. Every year, through photosynthesis, we capture about 121 billion tons of carbon dioxide, and we make 160 billion tons of carbohydrates. Heterotrophs are dependent on plants as a food source and for raw materials. It's like I keep telling you, even if you don't eat grass yourself, you eat something that eats grass. So without plants, we'd all be stuck like Chuck. Okay, this is my favorite part. All the solid material of every plant is built based on sunlight and out of thin air. All the solid material of every animal was built from plant material. So, everything you can think of, plants, cats, dogs, elephants, people, were really just particles of air woven together by strands of sunlight. Like I told you, when I do photosynthesis, I get a little philosophical, but it's the truth. Okay, hopefully this was simple and fairly clear. Let me know if you have any questions when I see you in class next week, and enjoy your weekend. Bye, guys.